Good evening and welcome. This is Reverend Marta Illueca from Brandywine Collaborative Ministries. And today I want to share a talk that I presented with a panel of 3 experts for the World Health Organization in Spanish for Panama and Latin America. But it was a talk and a panel that really gathered together the whole journey of a production of a vaccine for COVID-19, and I thought it would be wonderful if I could share it with my family in Delaware. That's all of you. So let me take you then through this journey. I'm gonna talk in 2 parts. I'm first gonna give you the general basic science background, and then we'll go through the actual process of the vaccine. So let me get to my PowerPoints. Okay. So the program was called COVID Global Race Toward a Vaccine, and my talk focuses on the journey from the laboratory bench to the clinical application to a patient or to us. I have no conflicts of interest. I am not doing any current work, nor do I get any financial remuneration from any pharmaceutical company. I did have a 12-year pharmaceutical career and consultancy with AstraZeneca Pharmaceuticals in Wilmington, Delaware, until the year 2015. I do not have any political party affiliation, and I am a clergy member of the Episcopal Church. I'm going to cover a few important topics. First, I will go through what a vaccine is, how does it work, what does it do, how does it do it? And then I'll get into the details of the phases of vaccine development and manufacture. So let's start with, I just want to share my excitement about the vaccine testing process. It brings together the main scientific disciplines that we are familiar with, you know, from basic science to chemistry, to immunology, to microbiology, to virology, and to clinical medicine, epidemiology, and infectious diseases, and then, of course, pharmaceutical drug development. It's a fantastic compendium of all these fabulous disciplines. Now, the World Health Organization has a very good definition of what a vaccine is. They call it a biological preparation that triggers an active or acquired immunity to a particular infectious disease. And you'll see the vaccines are active against only one infectious agent. And in simple words, it is really a protein that is alive. It has biological activity once it gets into the body, and it stimulates our immune system, the army of immune cells that we have, and it simulates, it's like a mock exercise, it's an attack. And by doing that, it trains our immune cells by provoking a specific defense against the attack. And for that, it will help in the production of antibodies. Those are these pieces that look like a Y. That's exactly the shape of an antibody, and these are proteins. And it also will help in the production of cells that will fight the infection that we're trying to protect with. Now, vaccines are not just produced against viruses. They are vaccines against bacteria. You remember DPT, diphtheria, pertussis, tetanus. It's an example. There are also vaccines against certain pneumonias and meningitis, etc. Vaccines against viruses have been around for a long time. You remember polio, measles, mumps, rubella. And now, of course, COVID, we're working on that. And then there's a lot of work going into creating vaccines against parasites. Now, parasites, because they are so big, and this is not a parasite, this is a mosquito that carries the parasite that causes malaria. But parasites are bigger organisms. And because of that, they're very complex. So it's not that simple to find a piece of the parasite that the vaccine can simulate inside the body. But I thought it would be good to remind us that vaccines work against a whole host of infectious agents. Now, COVID is a virus of the coronavirus family. That family was actually discovered in the 60s by a laboratory technician in Canada, a woman. And there are two kinds of viruses, but coronavirus specifically is of the type called RNA. So viruses are basically pieces of genetic material. And they come either as a double-stranded genetic piece, which is what we call DNA, deoxyribonucleic acid, and it simulates the same DNA that we carry. And then there are the single-stranded like COVID, where all we get is a piece of that genetic material that's called ribonucleic acid, RNA. And the little m means messenger, because whenever a virus gets into a cell, it has to talk the language of the cell. And it does that by this particle that needs to be produced that's called messenger. The COVID virus is very clever. He already has the messenger. He already has the dictionary to get in there and cause havoc and replicate. So this is sort of like a good diagram with all the different components of the virus. I also want to point out the envelope. The envelope of the virus has fat cells, and not cells, but fat particles. And those are the particles that get dissolved by disinfectants and by alcohol. And that's why it's so important. Wash hands, disinfect surfaces, because it destroys the cover of the virus, and that's it for it. And the other interesting thing about viruses is that they are not standalone agents. They are like computer programs that need a computer, or like apps that need a smartphone. So remember that. That's why the virus only becomes active inside us, because it needs a cell, one of our cells, to then do what it does, to make us sick and to replicate and replicate. Now, how does a vaccine work then? Well, let's figure out, we're talking about COVID. And by the way, SARS-CoV-2 is the actual name of the virus. Not very melodic. COVID is a little more melodic. The vaccine is like a secret agent that gets into the body, disguised as COVID. He's disguised as the agent that we need to attack. And it requires it's like a mask. Ironically, it also wears a mask. A mask that has those spikes. So corona means a crown, and the virus has these spikes. These are proteins that help it cling to the cell and enter the cell. So when the vaccine, when you get this molecule that is going to go into the body, it's going to have these simulated spikes, or is going to produce the spikes inside our body. And when that happens, that's going to sound an alarm in our body. And the alarm is going to have the army of the immune system respond. And we're going to have cells that come to the rescue. And we're going to have production of these pieces here that are antibodies. And antibodies look just like this. They are pieces of protein, and they look like a Y. So there's two lines of defense, the antibodies and cells. And some of the cell units are lymphocytes. Okay. Now, what about the types of vaccine, right? It gets confusing. There's mRNA here, there's live virus there's inactivated virus. There are quite a few of these platforms. So we call them platforms because these are the strategies to trick the body to create the immunity. So for example, you got these viral vectors. The examples used by University of Oxford, the collaboration between Oxford and AstraZeneca. In Cambridge, England, you also have, there's at least one Chinese company that is using also vectors. So vectors are like Trojan horses. They enter and then they release what's inside. And that's where the codification is to trigger the immune response. Then we got inactivated viruses also. And for inactivated viruses, we have quite a few Chinese versions of vaccines by Sinovac, Sinopharm in Beijing and Wuhan. We have other companies that are working just on pieces of the virus, virus particles, or even pieces of the protein of the virus. And some of the more exciting ones are actually spikes. So there's a platform that works just with creating the spikes of the virus and throwing it in. And there are a couple of these in Australia. Queensland University is an example. Vaccine is another example. And then we got Novavax here in Maryland. So that is another, another strategy. And then one of the newer strategies in the world of vaccines are the companies that are working with actually strands of RNA, okay? And example of that would be Moderna and Pfizer, and a couple of others are coming up. So that is a newer platform. So you see there's a whole host of types of potential vaccines, but always remember it's either gonna be about the RNA or the whole virus or DNA. So that's kind of like the variety of materials. So what I want to do now is I want to stop just for a few minutes because it's important, this basic science piece, this is at the heart of the discovery phase and the preclinical phase of the development of the vaccine. So if you have any questions about this, why don't we stop for 5 minutes and then we'll come back to talk about the actual production, testing and approval of the vaccines.